So we are going to introducing Professor Hugues Berry. So it's really a great honor for us having you with us today. It's a very important person in research in France and over. So Hugues Berry is a research director at INRIA, which is the French National Institute for Research and Digital Science and Technology. He was a deputy director of this institution, the Bush institution in France, between 2018 and 2023, as a deputy director in charge of digital health, digital biology, research in digital biology and health, and also several applications in neuroscience regarding the whole institute. So he is the co-head of this fantastic group called Astrocyte, which is a research group between the INRIA, but also, as you can see, Hospice Civil de Lyon, it's a hospital, and the Université Claude Bernard Lyon 1 in France. So the team is dedicated to develop innovative numerical method for neuropharmacology, mostly research in drug candidates to treat brain diseases. Hugues is a computational neuroscientist, and his research focuses on mathematics and computer models for studying the spatial control of dynamics of biochemical reaction involved in brain cells. One important work he did was modeling the metabolic and signaling networks at play at the level of the astrocyte. We talked about the astrocyte yesterday. We are going to hear more about what are astrocytes, what is the role of the astrocyte within the brain metabolism, but the brain activity too. I let right now Hugues presenting this first lecture, exactly as we did yesterday. So lecture of 1 hour and a half. Then we have like 30 minutes of break. We can also continue the discussion, and we come back for mostly a dedicated research program. So thank you very much for accepting our invitation.
Thank you very much, Habib, and thank you for the invitation. Thank you for the introduction first, very nice introduction, and thank you as well for the introduction. I'm very happy to be here with you today and try to talk about astrocytes. And really, I'm enjoying the workshop. So I'm really looking forward discussing with you and interacting with you. So this is more like a class and presentation of what are astrocytes and what they do in the brain, because at the end of the day, if you want to simulate or model a process, what you need is to know the process that you want to simulate. Yet it turns out that in biology, we still don't know very well what are astrocytes and what they do exactly. So I try to focus my talk on what are the main things that we know about astrocytes, like for sure, and what are the things that we don't know, and what are the main controversies, if you want, in the field. And there are many controversies in the field. So maybe it's going to be a bit too much of a biology for you. So if this is the case, don't hesitate to stop me and say, okay, I'm fed up with all these molecules and cells and so on. And of course, don't hesitate to interrupt me. It's better if it's a bit interactive, of course. So just to start with, I don't know if you know this story already, but this is a very nice story that began with Albert Einstein's death in Princeton in '55. So you can imagine Einstein was already the big genius and star and so on. So when he dies at Princeton, that's a small city, all of a sudden you get hundreds of journalists and people and fans from all over the world. So it's a bit a mess. And then within this kind of busy stuff, his pathologist, this guy called Harvey, Dr. Harvey, is here. It's him. Comes in Princeton to do the autopsy. So he does as usual. He takes the liver, examine the liver, the heart, and then he takes the brain and cuts it in big pieces. Puts the pieces in mason jars, so jam jars, and runs away with the jars from the hospital. And the guy, in fact, for 50 years, he will be hiding around in the US, moving around so that he's not recognized. And with him, keeping the jars with him with ancient brain in it. And it's been 52 years that he's been hiding. And this guy is even a book by a journalist telling his history in the country. And in fact, so by chance, Harvey died in 2007, and the brain went back to Princeton University as a donation. But why did he do that? In fact, he did that because he didn't want the brain of Einstein to be lost for science. I mean, if you just like burn the body or inhumate the body, he wants the brain to be useful. That means that quite regularly, he used to cut slices into the brain and send the slices to the best neuroscientists in the world. And in 1985, for instance, this woman, Marianne Diamond, received a small letter with a piece of a slice of Einstein's brain in it. And so she took the slice and she started to look at what's inside the slices. So first thing she does, she counts and she looks at the neurons. Well, the neurons, she finds no difference, nothing unusual. But then she does something that is very strange for the time. She looks at astrocytes. And she said, okay, the astrocytes look normal, but there are many more astrocytes in Einstein's brain than usual. And then, voila, genius of Einstein will be due to the fact that he has more astrocytes than the average. And I like this idea because it's a completely false paper. I mean, back at the day in 1985, it was very difficult to count the number of cells in the brains. And in addition, what she did, as a reference, she took brains from patients that were much younger than Einstein when he died. And it turns out that we know now that when you get older, the ratio between the number of astrocytes gets larger than when you're younger. So in fact, she didn't have the good reference. Anyway, so I think that this paper is wrong, but still I like it because it's the first paper that suggests that maybe the astrocyte will be something that counts when you look at intellectual activity. Now we go to, this was 1985, so 30 years afterwards, and maybe prove that, of course, the paper was wrong by Diamond, but she had a good idea, a good intuition, because this is a paper that a work that has been done by Michael Nedergaard in the US. And what she did, she grafted human astrocytes to mice, right? So the idea, you see it here, and we have human astrocytes in green and mice astrocytes in red. And in fact, human astrocytes, they are much bigger, 3 or 4 times bigger. They are much more complex with much more branches and ramifications and so on. And the idea will be, are we human beings more clever than mice? Depends on the human being, maybe, but because we have bigger and more complex astrocytes, right? So she grafted the astrocyte to mouse, and she put the mouse to, behavioral tests. And the thing she did a test of noise shock. So she got the mice, and she gave an electric shock to the mice coupled to sound. And then afterwards, she gives only the sound, and the mice must learn that the sound is associated to the shock, and it must freeze, right? And what she discovered is that the mice that were grafted with human astrocyte were much better. The percentage of them that were freezing, that had understood that the noise was coupled to the shock, were much bigger than those that were not grafted. Of course, she said, okay, maybe it's only because maybe it's only because the astrocyte allows you to hear better. So she did the same with stimulus that were not auditive, like visual stimulus, and it was the same. So it was a very nice paper that showed that maybe, again, there is something with astrocyte related to intelligence and not just metabolism. And a last example also is more recently. This is about depression. So this is something that tells you how that astrocyte can be implicated into pathologies as well. So it was a paper that we did with my colleague Ailan Hu in China. In fact, I mean, we are co-author of this paper, but really at the end of the day, there's almost nothing that we did in the paper because it was pushed the supplementary material. But what she did, she wanted to look at mice that were models of depression. Okay, whatever you can think what you want about, I mean, animal model of depression. Still, what they did to test whether the animals are depressed, what you do, you put them in the water, and you look at how long they are swimming. And the mice that are depressed, they stop swimming much faster because they are depressed and they say, okay, what's wrong? I mean, I don't care. I don't want to live. It's better to stop swimming. Well, at least that's my interpretation of the experiment. And so if you compare the duration time during which the mice remain immobile and stop swimming in the water, you see that the control mice, they don't stay immobile for long, and the CLH, the blue ones that are the animal model, they stay immobile for twice as long, right? So that's how they measure the symptom of depression, if you want. And so what they did, they look in a small part of the brain that goes the lateral habenula, and they took all the astrocytes and the neurons, and they tried to compare with proteomics what was the difference, what molecules were, or RNA, no, that was proteins, were more expressed than the other. And what they found that the main difference was due to a small potassium channel, the KER 4.1, that were much more expressed in the depressed animals, that's rats, not mice, sorry, than in the non-depressed ones. And they were not highly expressed anywhere. They were highly expressed, the KER 4.1, in the astrocytes only of the lateral habenula of the depressed animals. So yeah, that's just correlation. So what she did, and that was a very nice, it's one of the few examples where biologists are able to look at causality and not only correlation. What she did, she took, and that was Irwe Kui did while she was a postdoc there. So they took control animal, and with gene technology, they overexpressed the KER 4.1 in the astrocyte of the habenula of this control animal. And they put them to the test of depression, and they observed that the gain of function led this animal to have symptoms that are like the depression symptoms. So if you want, you overexpress KER 4.1 in the astrocyte, and normal animals becomes depressed. And conversely, you can do the reverse. You can take your depressed animal and remove the KER 4.1 of the astrocyte, and again, they become like normal animals, right? So, of course, that's only, this doesn't mean that it works like that in humans, right? Because it's an animal model. But at least in these animal models, KER 4.1 is alone sufficient to explain the depressive symptom. Right, so this will be the outline of my talk. So I will give you an example, a list of what are the consensus currently of what astrocyte does, what they do, and then a list of controversies and things that we don't know yet. The first thing will be what we don't know is about the calcium signaling. You will see that most of what astrocytes do, they do it because of calcium signaling, but still we don't understand very well how it works, right? And then other controversies on the function of the signal and also other roles that we can talk about together. And as a perspective at the end, I will give you some ideas of how can we do to go from intracellular signaling to network models, because there's still a long way to go. Okay, so what is the element of that everybody agrees on in the community? First thing is that there are astrocytes in the brain, that's for sure. Question is how many? I mean, what is the ratio between neurons and astrocytes, and even the neurons to glial cell? Because you can read, it's fun. This paper here explains. You can see sometimes in the introduction of papers that the glia to neurons ratio is more than 10. But there's no, in fact, there's no experimental claim for that. I mean, experimental support for that. In fact, if you look at really the real paper, you see that the ratio between the number of glial cells to the number of neurons in the brain, of course, it varies quite much depending on where you look at in the cortex or in the basal ganglia and so on. It depends on if you look in the white matter or in the gray matter and so on. But it's around 1:1 to 3:1. So at most, you can find 3 times more glial cells than astrocytes. So glial cell is astrocyte plus microglia plus oligodendrocytes. So in fact, synapses are 20 to 40% of the 3 of them. So you came to a number, an estimation of astrocyte to neurons ratio that varies between 2:1, so twice more astrocyte than neurons, and 1:3, that is 3-fold more neurons than astrocytes. Again, that depends on where you look at. In humans, I'm saying, right? These numbers. So it's something around this number. So that's almost accepted, right? Now the roles. There are many roles of astrocytes, and we'll discuss about that in a moment. Astrocytes are very important for brain development. They are very important in terms for the brain because they are parts of they are implementing the blood-brain barrier, at least some part of it, because there are some ideas and maybe parasites are as well active. They are very important for homeostasis, and we will come back to that for calcium, potassium, and glutamate, for instance. They are very important for the metabolism of the brain. They are very important for the repair of injury and for information processing. So for that, everybody almost agrees. The idea is, why are they so important, and what are the mechanisms that are implied? The main mechanism is the so-called tripartite synapse, right? So in fact, astrocytes, they have their cytoplasm, their plasma membrane, they have extension, their branches that becomes smaller and smaller. And from time to time, they can reach and touch a synapse, and then wrapped around the synapse. You have an example here with the electron microscopy, the green with the vesicles, the presynaptic neurons, the yellow with the PSD is the postsynaptic, and the blue here is the astrocyte. The astrocyte enwraps around the synapse. So when you have in this kind of configuration, the neurotransmitter that is released when the spike arrives there. So of course, the neurotransmitter will bind to the postsynaptic, of course. But it turned out that the astrocyte, they had all the equipment to bind the neurotransmitter as well. They have glutamate receptor, they have GABA receptor, they have acetylcholine receptors, and so on. So they can bind to these receptors, and they will react just like the postsynaptic neurons by a transient of calcium, cytosolic calcium. So you will have a spike of calcium, a peak of calcium, when they receive the neurotransmitters. But contrarily to the postsynaptic neurons, the astrocytes, they don't have active electric signals. So they don't do action potential. So they can do depolarization, but they don't do spikes, voltage spike. What they do is calcium signals. And that will be the main point of the rest of the talk, is that the activity of the astrocytes are or seems to be encoded in the calcium signal of their calcium content, cytosolic calcium. Also, and we will go back to that in a moment, it can be that in a calcium-dependent way, some of the astrocytes are able to release themselves neurotransmitters, or we call them gliotransmitters because it comes from the glia, but it's the same ones. It's glutamate, GABA, and they can release it as well. And of course, if they can release it in a calcium-dependent way, it will, of course, modify the synapse. So these tripartite synapses are very important. Now, in human, in fact, a single astrocyte contacts around 2 million synapses. One astrocyte contacts around 2 million synapses. It's 10 times less in rats or in rodents, but it's a huge number of synapses that is contacted by a single astrocyte. And if you take in the cortex at least one synapse at random, it depends on where you look at, but you have between 30 and 90% probability that this synapse is tripartite. So you have part of the brain where almost all the synapses are tripartite, another where it's only 1/3 of them. So I come back to say the idea is, of course, that calcium activity is the main activity of astrocytes. So you have a really nice movie here. This is a fluorescent measurements. So calcium gives fluorescent signal, and you see the signal here. And what you see is the nice. This is in vivo. You see the cell here, the cell body of the astrocyte, the main branching astrocyte, and the small domain there. And you see all this oscillates. So all these are calcium signal in the astrocyte. And the main important idea is that when you stimulate an astrocyte, for instance, if you release glutamate close to there, what you will see in terms of calcium signal, so this is the calcium signal, is not an increase of calcium. It's oscillation of calcium. So somehow the transduction is made on oscillations. And those are not simulations, those are real measurements, for instance. The main issue is that astrocytes are, in fact, not at all stars. I mean, astro means stars, but they are not stars, because in fact, their shape is very complex. So you have, again, the cell body, the main branches, and that's what is difficult to see here. What you have is the main branches, they divide into very smaller and smaller branchlets and leaflets, and they are so small that they are below the diffraction limit. So with conventional confocal microscopy, you just see this mist of signal, right? Because it's too small, but they form very small branchlets. And if you look at this branchlet, I love this movie that's been done by our colleague in Bordeaux, Valentin Hegel. So what you see here is a dendrite in pink, and that's the bouton. So the synapses are there. That's the postsynaptic boutons, and the white shapes are the plasma membrane of the astrocyte, right? So what's inside the white stuff, contour, is the astrocyte. And in blue, what you see are the local calcium signal. And I will play it again. And what you see that you have sometimes calcium signal that comes into places like there where it seems that there are no synapses. So that means that you have intrinsic calcium signals, key signals. But most of the time, when you see a calcium signal, it comes there, for instance, where you have a dendrite or a synapse not so far. For instance, you see here, you will see calcium signal there that comes very close to the synapse there. So those thin branchlets that you see here, that make this what we call the glia peel, so the very small part of the astrocyte. They are very small, like less than 40 to 50 nanometers. It's very thin, but they make up more than 3/4 of the total cytoplasm of the astrocyte. That means that for very long, people in the community, they have just looked at the cell soma and the main branches. But if you look only at the cell soma and the main branches, you only look at 25% of the astrocyte. And in terms of calcium signals, they make up more than 80% of the total calcium signals. That's where the things happen, it seems. Of course, the astrocyte, they also interact with vessels, blood circulation, via specialized structures that are called NFIT. For instance, again, it's a question of calcium signaling. And here we know that when the astrocytes control the dilation or the contraction of the vessel, and it does that with some kind of calcium. At least it's correlated to variation of the calcium signal in the NFIT as well. So calcium signaling in the NFIT also is somehow related to vasodilation. Vasodilation, sorry. And of course, this is related to fMRI, because this is what the so-called neurovascular unit, and this is where the fMRI signal comes from. For instance, you see this very nice paper there, where people have been able to do both a measure of the bold signal locally and of the calcium signal, so the spikes in the neurons nearby and in the astrocytes nearby. And you see that, of course, turned out the bold change, the bold signal is quite well correlated to the neuronal activity. I hope it's the case at least. But if you look at the astrocyte calcium activity, you will see that they are as well very well correlated. So at the end of the day, when we think that the bold signal tells you something about the neuronal activity, maybe the bold signal tells you something about the astrocytic activity. Nobody knows really. Well, this is a very nice paper. If you want to look at it, you will see there are some subtleties, but it's more subtle than that, but still there is a correlation. And it's not also astrocytes, they also form networks. They are interconnected, they interconnect to each other. That's very nice rainbow-like stuff with astrocyte. Each color is a different cell, a different astrocyte in a mice brain. And they are interconnected by gap junctions, so the grouping of connexins, connexin 13 or 43 for the astrocyte. And through those connexins, they can exchange IP3, we'll come back to that, glutamate, ATP, glucose, lactate, glutamate, whatever, ions, potassium, for instance. So you can have communication between the astrocytes. And for a time, people were very excited about intercellular calcium waves, because if you do in vitro culture, you see many waves of calcium going from one astrocyte to the other, right? Because you can have this kind of passing of message of calcium reaction diffusion waves. This was immensely observed in vitro, right, in cell culture. Now, does it exist in vivo, those calcium intercellular waves? It's not sure. When people look at those waves in vivo, usually they fail to see them. Well, at least at the current state of the things. Okay, so why is it so complex? It is so complex because still, so that's the main picture that I gave you. Now, if we look in detail, there are many, many things that we still don't understand. And in particular, there are many things that we don't understand in the calcium signal itself in the astrocytes. First thing we don't know is that where does this calcium come from, right? Of course, you see in the cytosol, you see calcium transients, it changes. Where does it come from? Does it come from the outside of the cell? So if it increases, what seems a bit acknowledged is that most of the time, but not always, it comes from intracellular stores, calcium stores. For instance, the endoplasmic reticulum. It can be also the mitochondria. It can be the lysosome, for instance. But still, most of the time, it's the endoplasmic reticulum. So the main idea is that this is the scheme, this is how it happens, usually. When the postsynaptic neurons fires, for example, you have in the astrocyte, for example, the metabolic glutamate receptor that binds the glutamate, for instance. This will create new IP3 small molecule. And this IP3 can bind to this IP3 receptor channels at the endoplasmic reticulum. And when this IP3 receptor channels, that I will come back to in a moment, when it binds IP3 and one molecule of calcium, it can open. And when it opens, it's a calcium channel. So the calcium can go from the reticulum, endoplasmic reticulum, because it's the gradient is in favor of exiting, and it will go in the cytosol. So that gives you the surge of calcium going in the cytosol. Turns out that this guy, the IP3 receptor channels, it also closes when calcium here is large. So if you have a surge of calcium, this opens, and it will also close the IP3 receptor channel. And we will come back to that in a moment. And then you have an ATPase called the Cerca pump that just consumes one molecule of ATP to take the calcium there in the cytosol and reintegrate it in the ER. So that's what the main picture. And in fact, that's what happens in the soma. And this is a very nice paper that shows that in the soma, if you look in the soma, the calcium signal, that is this one, it's exactly controlled by that. I mean, nothing comes, almost no calcium comes from the outside. And the proof of that is that if you mutate this guy, you get rid of this guy, like IP3 receptor channels, that's what it does here. In the soma, you mostly have no calcium signal. But again, beware that this is only 25% of the cell itself. Now, if you look in the microdomains, so small branchlets, the very small branchlets, that's where the interaction with neurons happens. That's very different. First, the first thing you see that this is in black, what happens here in the red or yellow stuff. You see the calcium signal is very different from the soma. It's much smaller. The time scale is much faster. I mean, the peaks here are much are less width. They are less longer. They less long than in the soma and so on. And then in there, if you mutate the IP3 receptor there, then you keep the almost the same amplitude. The amplitude is decreased, but the frequency of the calcium signal is identical. That means that in those small branchlets, most of the signal doesn't come from IP3 receptor channel. So it comes from elsewhere, but where we don't know. It could come from some plasma membrane channel, calcium channel, but it's still unidentified. It could be that it's a question of IP3 receptor, that there could be subtypes of IP3 receptor that are not mutated by this. I don't want to go into too much detail, but at least that means that we don't know yet in the branchlet, we don't know for sure where the calcium comes from. So that's a bit, if you do modeling, if you want to do a mathematical model of this kind of system, if you don't know where the calcium comes from, it's a bit of a pain, right? Anyway, up to now, so just to say that this result is this kind of idea that we don't know what happens really in branchlets in terms of receptors is a recent realization. It's 5 or 6 years that we are aware of that. Up to now, people were absolutely sure that what happened in the soma was the same as what happened in the branchlets. So the first mathematical models, and I did that as well 15 years ago, where we started with this kind of system because we were sure that it was what happened. So the idea is that this guy, the IP3 receptor channel, is a very weird animal, if you want. So it's a calcium channel. So when it opens, it transfers calcium from the ER to the cytosol. But this is a very nice experiment when you look at the open fraction of the channel as the calcium concentration increases in the cytosol. And this is, well, I won't go into too much detail, but it's an equilibrium concentration that the open fraction that you see here and the fraction that you see where you average over a large population of receptors and very long time the open fraction. And you see that when you start to have a bit of calcium, it will open the channel. But when you have large amount of calcium, it will close the channel. So that's this bell-shaped curve, right? And so once you have that, you can start to do a model. So that's what's been done by De Jong and Kaiser 30 years ago already, 35 years ago. And it's just a Markov, a simple Markov model. So the idea, but this we still don't know if it's true in terms of molecular structure. The idea behind this model is that the model assumes that there are 3 binding sites in the IP3 receptor channel. 2 for calcium. One is activating and it has a high affinity. The other one is inhibiting, so it closes the channel and it has a low activity, right? So this one will be active when you have a small number of small concentration of calcium, and this one will only be active at large concentration of calcium. And then you have also a binding site for IP3. And the open version is when you have 1 calcium on this site, 1 calcium, 1 IP3 bound on this site, and nothing in the inactivating channel, right? Binding site. So the open configuration is a 1, 0, 1 if you want to write it like that. And then it's a Markov model. So this is the state where you have bound 1 active calcium, 1 calcium in the active site and nothing else where. And if you bind 1 IP3 there, you go to the 101 configuration and so on and so on. So you can do a very nice model of that. And that's what's been done here by De Jong and Kaiser. And at the end of the day, what you have is, in total, you have 7 ordinary differential equations, 1 for each of the state, except that it's a fraction of, since the number of states is conserved, you can remove one equation. So very simple, it's a law of mass action. I won't go into detail, but it's a very linear way of writing the chemical kinetics. And we also have, of course, one concentration for calcium, because that's what you're looking for. And what the model tells you here is that the open state is this 101 state. But to be open, you have to have this exponent of 3 there. So that means that you have to have 3 channels that are clustered that opens simultaneously. So it's cooperativity. So that's a nice model, but it's still 8 differential equations. So sometimes afterwards, Rinzel came and used a slow fast time scale separation. So that's a very usual stuff that we do in under ordinary differential equation model. If you consider that some variables have a time scale that is faster than others, you can separate the two, and this you can simplify the equation. That's what they did, and Lee and Rinzel did that. So at the end of the day, they come with a model where you just have 2 variables. One is for the calcium, it's the same as before, and one is for age, and age is just a fraction of the sites that are not bound by the inhibitory calcium. So age is a fraction of the receptors, the number of molecules that can be closed, and that is not closed yet, right? And if you do, so this is a very nonlinear ODE, because, for instance, you have this edge there that has a power here of n edge, and this gives you the nonlinearity. And since it's nonlinear, typically what you have is that if you look at the IP3 concentration, you will see that sometimes you go into Hopf bifurcations, right? So that when you increase the IP3, in the model IP3 is constant, it's just a parameter. And if you increase the IP3, you will go from a point there to there when you will cross 2 Hopf bifurcation. And in the middle, what you will see is oscillations, right? And if exactly what happens here, this example there, you start at a small concentration, IP 0.2 micromolars IP3. So you have a stationary state, you're stable. And then when you go there to 0.5, then you will run into the oscillatory, and we go with the oscillations. And of course, this is nice because, again, what you see in the astrocyte are oscillation of the calcium. The main problem with this model is that it's very sensitive to the exponent. So this is NH, this is a parameter that gives you the exponent for the nonlinearity. And if you decrease NH from 3 to up to 2.6, that's not much. And then you lose the nonlinearity, you lose the Hopf bifurcation, you lost, and then you don't see the oscillations any longer. So that's a bit of an issue. But we'll come back to that later. Now, okay, so we sort of understand we have this IP3 receptor channel. A model of this channel gives you hub bifurcation. Hub bifurcation, they tell you that you can oscillate, and this gives you the peaks that you see and the spikes of calcium that you see in the model. Now the question is, and this is a hugely important question in the field, is what is a calcium transduction? Meaning, if you increase the input, for instance, if you increase the glutamate level, or if you increase the frequency of neurons stimulation, how will you what will be the change of the calcium? So the input, the glutamate, or the fraction of the rate of the presynaptic neuron are the input, and the output is your calcium. So what do you expect if you increase the stimulation? Do you expect that you have more calcium? No, because you have oscillation. So that's not a dose response type of stuff. So you have oscillation, but what will change? Will you change when you increase the input? Will you change the amplitude of the calcium oscillation? Will you change the frequency? Typically in neurons, in neurons, the action potential, they are supposed to be elementary, so it's the same amplitude. So what you change you expect to change when you change input in neurons, you expect to change the frequency of the spikes. That's the so-called frequency modulation. What is it for calcium in astrocyte? Nobody knows. It seems a bit weird, but we don't know. And in fact, the only papers that have started to look at that, they are very old papers, like almost 25 years ago, and it's not clear cut. You see an example here. So this is the fluorescence, each trace in black, that the calcium, it's something that tells you of the calcium in the astrocyte. And of course, this is in the soma. Again, you know that in the soma, I don't know what, but it's in the soma. And this is what happens when you have a small, when the astrocyte, then the neurons, they fire at a low frequency, 0.2 hertz. So you see you have in the astrocyte those peaks that are with a small frequency. And when you increase experimentally the firing frequency of the presynaptic neurons, what you see, you have both increased frequency. Well, the recording is not very long, so it's difficult to say, but you also have increase in amplitude, it seems. And that's the same kind of measurement. And now it seems that you have a change in frequency, maybe a change in amplitude as well, not clear. So my point is that we don't know. So at least can we have a model that gives you this kind of behaviors, that can choose between the two behaviors? And that's what we did, gosh, 24 years ago, no, 15 years ago, better. 15 years ago with Maurizio De Pita. Maurizio, at the time, he was a master student in Tel Aviv University with Eshel Ben Jacob, and Maurizio is now in Toronto. He has a permanent position in Toronto. And so we took the same model. So this is a Lee-Rinzel model, the same one that I showed before with calcium and the age. And we said, okay, but the IP3 in the Lee-Rinzel model, the IP3 is a constant, is a parameter. Yet in reality, in the cell, IP3 is a molecule, so it's been degraded by some enzymes. It's been created by other enzymes. So we just added the process of IP3 synthesis and IP3 degradation. Right? I don't want to go into too much detail, so how we did that, but you see in particular that some of the processes that produce IP3, they are themselves calcium dependent. So you have a strong coupling between calcium and IP3. And when we did that, well, in fact, so now we can look at what happens to the calcium with the glutamate here increases. And when we do that, most of the time we see that for the calcium, we have this kind of bifurcation diagram. So it's a typical hub bifurcation. You have two hub bifurcation there. So you have a fixed point there and a fixed point there. And in the middle, you have oscillation, and the dots there gives you the amplitude of the oscillations. And what you see here is that you have what's called a supracritical bifurcation. And because it's supracritical, you have almost no change in amplitude, only a change in period there, also in frequency. That typically, with this kind of parameters, you get a FM transduction, frequency modulation for the calcium. But if you look at for the IP3, you see that it's both frequency, and amplitude. So it's AFM. Tell me if it's not very clear. Just don't hesitate. But of course, if you have a model like that, most of the parameters, not all, but many of them, you don't know them, what are their real values. So if you change a bit some of those parameters, you can have other regimes, for instance, this scaffold regime, where the hope with bifurcation here is no more supercritical, it's subcritical. And in this case, when the limit cycle is born here, you see that its amplitude increases slightly with glutamate concentration. Whereas here, when it burns, when it's born, the limit cycle here has already a very large amplitude. So in this case, with those parameters, even for calcium, you have a transduction that is AFM, and that's not only FM. Right. So of course, I would love to find experimentalists that will get back to the benchtop and try to determine what happens in the astrocyte. Is it really AM, AFM, FM? But we don't have this data, and that's really that's an issue for us, But at least you can have, if you add, compared to the Lee-Rinzel model, if you add the dynamics of the IP3, that you can have some things that are more complex and that can have more complex behaviors. And then at the end, and we'll stop with that for the calcium signals, the main issue with this where is it? This Lee-Rinzel model. The first model there is what the experimental data that were used to calibrate it were very long equilibrium data. So that where you compute the open fraction, you measure the open fraction that you average over a large population of receptors and very long times. Now, since then, we can do, with patch clamp, we can do single molecule recordings. And that's much better, because now you have this kind of experiments where you measure the current, so the open fraction of a single AP3 receptor channel. And what you see is that this range of, like many receptors, it oscillates spontaneously between a state where it's closed, the current doesn't go through it, and a state where it opens, that's where you have here, you have minus 50 picoamperes current when it opens. So the stuff, the single molecules oscillate between close, open, close, open, close, open. And what you see when you do the recordings is that you have two types of regime. A regime there where most of the time the channel is in the closed configuration. From time to time it goes to open, but usually it remains closed. And those kind of period there when it will go to the open state much more often, right? And the authors who did this, they call this a park and drive model, just like when you drive an automatic car, right? When you are in the park site, you don't move. So most of the time the channel is in the closed configuration. And when you trigger the drive position, then you are much more likely to go to the open configuration. So once they have that, when you can start to do Markov-Chain models with different type of open and closed configuration, you can use Markov-Chain Monte Carlo to simulate it and to try to do model selection to guess, given a collection of possible models, what is the best model that explains your data the best. And what they found, and that's a very nice series of papers by Zickman 10 years ago, that in fact the simpler and the best model to explain that is this model when the park state has two configuration, one closed and one open, and the drive has three closed and one open. And where calcium comes into play, calcium will only trigger or increase the probability to go from C2 to C4, so from drive to park, and C4 to C2, right? So if you don't have much calcium, the probability to go from C2 to C4 is very low. That's a green signal there. Whereas the Q24, so the probability for C4 to C2 is very large. So you are mostly in the drive case, and in the drive configuration, this is very small. So it's very unlikely that you go to the open. So you remain closed most of the time. And when you have an intermediate number of calcium concentration calcium, it's the other way around. Q42 increases, the Q4 decreases. So you go there and you swap from one to the other, basically. This is a very nice model published 15 years ago, and nobody has ever used it in a model of calcium signaling astrocytes. And honestly, I still don't know why. I can imagine why, but it's a very, very nice model. Unfortunately, it's not been used very much. Ivo Zickman is now in the UK, I think. He has a lab in the UK, but it does something else. And I don't know why this model is not used. Okay. Now, so this was about this calcium signal, how it is built, what it does. Now the question is, what is the function of the signal? So you have calcium, the calcium that does peaks and so on. What it is used for in the astrocytes? And the first hugely important question is propagation and integration. My point is that, so you have a calcium signal. Let's say that at your tripartite synapse here, you have a calcium peak that arises there. And what will happen? Is it like neurons? Do you expect that this calcium signal will be propagated down the down the branchlet to the soma? Of course, that's the reflex of any neuroscientist. They will say, yeah, it works like in neurons. But is it true? And then if you have two calcium signals coming and meeting each other, like when you have two EPSP in neurons in the dendrite, what will happen? Will they add up linearly, non-linearly? We don't know. Nobody knows. And that's really an issue, right? And in fact, it should have been clear that it's an easy stuff, because as soon as 10 years ago, this very nice paper by Balkak. Balkak is a very nice experimentalist in Los Angeles, working on the astrocyte. And what he did is this very nice measurement of calcium activity of an astrocyte. So you see the cell body is here, the main branches is here. So all this gray stuff is the branchlets. And here you see two end feet. So you see the vessels and the end foot that is there and there. And you measure simultaneously calcium activity on all those compartments. And what you see in green, that's what happens in the branchlets or the main branches. You see large calcium signal. And in red, that's the calcium signal in the two end feet. And what you see that it seems that there's no correlation at all. The same cell, when you have a huge peak of calcium there in an end feet, but nothing there. Maybe, well, this is just one example, one short example. But when you look at the recordings, you see no propagation. You see no clear correlation between the stuff. And that's even worse. And that's even worse because this is 2D, right? This was a 2D recording because that's what people can do. But the problem is that when you see, for instance, let's say that you have a wave, a calcium wave. So let's say the calcium is transmitted. If you see starting here and stopping there, you say, okay, the wave stops. But still, you only look at a slice. Maybe it goes in the third direction and you only missed it. So what you need to be sure of this propagation is a 3D monitoring. And that's usually, technically, that's usually difficult. Andrea Volterra in Genève, in Lausanne, sorry, started to do that or proposed something like that 5 years ago. So he looked in 3D, sort of 3D. We can discuss about that if it's native 3D or not, and how it is propagated. And by the way, what you'll see, I didn't show you that, but what he proves is that if you look at 3D, you have threefold more calcium signal than in 2D. So you lose in 2D, you lose mostly three-fourths of your signals. So he looked at all that, and what he found was that if you look at calcium signals that are born in the processes, right? So out of the soma and the end foot, in the branchlets and the branches, 80% of the time, so almost all of them, they are local. So they don't propagate to the cell body. They just go there, they live there, and they die. They don't go up to there, 80% of them. In the end, food is the same. I don't know, I don't remember the number, but most of the signals, the calcium signals that are born in the end food, they remain in the end food. They don't go out of the end food to go somewhere in the cell, at least in calcium. Occasionally, so it's very rare, but it happens, he sees signals that originate from the cell body, he calls them global signals, and invade the whole cell. So you see some rare signals that are born there in the cell soma and invade everything. But most of the time, it remains in the soma. So that's very recently, and it's a paper that's been published by Kenneth Murray's group, just close to here in McGill. It looks like it could be something that is related to the morphology of the astrocyte. So what they did electron microscopy of astrocytes, and they went really to see into the small branchlets, very small ones. This is easy to do. This is easier to do in electron microscopy because you can go in a very small spatial resolution. And they looked at the shape of the astrocyte processes, and that's the yellow stuff, and in green you have the synapses. And what he saw, and what they saw, is that most of the time you have this kind of clusters in green, clusters of synapses that are grouped around a small, kind of a bigger part of the astrocyte. So this is very new, right? This was published 2 months ago. This means that maybe this seems to say that the synapses, they could be grouped into clusters, and those clusters of synapses there will share somehow the same calcium signal, but the calcium signal would not be shared between synapses from 2 different clusters. Well, again, this is only electron microscopy. There were no calcium measurements at all in there, and here it's only calcium. So it's an open question, but maybe that's a sign of a direction where the domain will go in the future. So only local, most of the time you only have clusters of synapses that comes from many different neurons, but that share the same calcium signal and that are isolated from other synapse clusters. So why is it important? Because of course, it's important because on top of that, you have gliotransmission. So again, gliotransmission is this idea that just like in presynaptic neurons, in the astrocytes, when you have calcium elevation, this calcium elevation could give rise to the release by the astrocyte of glutamate, glycerin, GABA, ATP, so neurotransmitter. And of course, this will impact on the presynaptic and on the postsynaptic neuron, right? And if you can look at it in the literature, so of course, some astrocytes, they have NMDA receptors in the synapse. Some of them have NMDA receptor or metabotropic glutamate receptor extra in the close to the synapse or presynaptic. So you have this. You can have this kind of receptors: glutamate, ATP, GABA, everywhere in the presynaptic and the postsynaptic. Depends on the synapse. So that means that, of course, if you look at all that, so you can find paper where you know that when you kill the astrocyte, you change the excitability of the postsynaptic neurons, meaning that, of course, you have experimental data where people have considered that astrocytes can act as an increase to increase the postsynaptic neurons excitability, and this is mostly due to those guys, the perisynaptic NMDA receptor in the postsynaptic, because when the astrocyte will release glutamate, this will depolarize the postsynaptic astrocytes, the postsynaptic neuron, right? Sorry, with what's called slow inward current, and for long, like it lasts 1 or 2 seconds, the postsynaptic will depolarize, so it will be more excitable, of course. But you have also many other kind of impacts. Astrocytes have been shown to impair short-term plasticity. We'll come back to that. They have been shown to mediate long-term, like LTP, LTD, so that long-term plasticity, so synaptic plasticity, so this is memories, right? And learning in the hippocampus, heterosynaptically or homosynaptically, and so on. They also have been shown to impact cortical SDP, so short-term, many kinds of plasticity have been reported as being impacted by astrocytes. And if got, it's not a wonder, because if you look at all the possible interactions that are possible between astrocytes, presynaptic, and postsynaptic neurons, of course, if you perturbate any stuff in this very complex network, you expect to perturbate the functioning of the synapse and the plasticity. It's no wonder, right? So the question is that, of course, all the synapses are not equipped with all these kind of receptors. Some synapses have this and this, but not that. So it depends on what type of synapse exactly you are looking at. But it depends maybe on what type of astrocytes. And something that we still don't know is there something like a Dell's rule for gliotransmission? I don't know if you know what is the Dell's rule? The Dell's rule is what tells you that in neurons, you have segregation of the neurotransmitter, meaning that neurons, some of them release glutamate, some of them release GABA, but the ones that release glutamate, they only release glutamate. All the buttons will release glutamate, and the ones that release GABA will only release GABA, right? Is it the same for astrocytes? We don't know. We know that astrocyte can release glutamate, can release GABA, but is it possible that the same cell release both glutamate and GABA, and where? At the same perisynaptic process, we don't know. By the way, this Dell's rule, of course, we are all used of that, but it's true that it is valid for most of the neurons, but it's not for all the neurons, meaning that we have neurons, we have examples of neurons that release both glutamate and GABA, sometimes in two different types of vesicles, presynaptic vesicles, and sometimes in the same vesicle. Some neurons in the same presynaptic vesicle, they have GABA and glutamate. So this Dell's rule is not true for all the neurons. For astrocytes, sadly, we still don't know. And again, for modeler, if you want to build a mathematical model, it's difficult not to know that for you. Of course, we can explore that, and we can explore, since you have so many different possibilities, like it's like, you know, it's like a puzzle. You can say, I will, let me take a bit of glutamate release and DC-RINE release by your astrocyte, and then in my pre, I will have NMDA receptor and peer to P2Y1 receptor and so on. So you can try to play into what is possibly what is possible. And this is an example that we did again with Maurizio some times ago. So we said, okay, so we have a presynaptic in blue that releases gliotransmitter like glutamate. We have a postsynaptic terminal that gets it. And we have the astrocyte in green that can bind glutamate, do calcium signals in response to this glutamate, and release again glutamate to the same presynaptic or to another presynaptic neuron, right? So if you want to model that, so we get back the thing that we saw before. So we still have this model. This is again this IP3 receptor channel model that I've shown you some slides ago. This is the same one. We plugged it with a presynaptic model. This is a model for short-term plasticity. Short-term plasticity most of the time is due to the fact that, at least that the so-called SODIX Urkram model, when you release glutamate, your number of vesicles is limited, right? So if you decrease, if you release much glutamate, huge numbers, you will deplete your presynaptic terminal from the vesicle. So if you have a new action potential coming in, then the action potential will see less vesicles coming in, and then you will release less glutamate, so you will have smaller EPSP, right? So that's where this short-term plasticity comes from. So this is only due to the recycling of the glutamate of the presynaptic neurons. So at least we had a model of that, and we just added a third model that is given the calcium that we have here in the astrocytes. Then we will do a release of glutamate from the astrocytes. And this is the same model as there. So it's some idea of you have glutamate vesicle in the astrocyte, and they can be released. I don't want to go into too much detail. We can come back after that if you want, but it can go once you get above a threshold, when the calcium becomes larger than the threshold in the astrocyte, you will release the glutamate in the synaptic cleft, and this glutamate, it's almost the end, can bind this time the tau variable to the presynaptic receptor. And then when it binds to the presynaptic receptor, what does it do? It will change what happens there. Again, I want to go into too much detail, but that's a very simple. I mean, there are 1, 2, 3, 4, 5, maybe 6 or 7 differential equations, but it's a very simple model. And what we saw is that, in fact, with this when this kind of stuff is taken into account, what you change, you will change the short-term. The astrocyte can change the short-term plasticity of the synapse. So let me explain that. This is what happens without astrocyte, and this is a raster plot in the simulation. So here, from 1 to 100, you have 100 presynaptic spikes, right? It's just a Poisson train. And it goes from this blue presynaptic stuff. And here you have a case where, because the timings of the recycling of the vesicles and the frequency of the presynaptic spikes, most of the time, when you have a new spikes coming in, it will release more glutamate than the previous one. Right? So the form of plasticity that is called short-term facilitation. And each time that you have facilitation, the spike there is shown in pink, whereas the other one, when you have short-term depression, meaning that the spike, the next spike is release less glutamate than the previous one, it's in green. And you see that there, typically what we have is a synapse with the time scale that you use, the parameter that you use, that has a potentiation, meaning each time you have a presynaptic spike, it releases more glutamate than the previous one. Now, what happens here is that the picture that you have when you add the astrocyte, that's the triangle here gives you the moment where the astrocyte will release glutamate. And you see that as soon as the astrocyte releases glutamate, everything becomes green, which means that now the synapse, because was facilitating, now it becomes depressing, meaning that each new incoming spike releases less glutamate from the previous one. So that is a very simple, and we can do theoretical calculation that explains why the astrocyte can swap the synapse from short-term depressing to short-term facilitating. And there you have the other case where you have a synapse that is initially in a short-term depression configuration, and when the astrocyte release glutamate fires, you have short-term depression. So that means that's something that you can use to understand how the astrocyte can change this stuff. And recently, Nicolas, Brunel, and Maurizio, they use very same ideas, this time for working memory. So there's the same kind of model, except that they were, in this case, the homosynaptic case, if you want, meaning that when the astrocyte releases glutamate, it releases glutamate to its own presynaptic terminal, not to another one. So it's a looped scenario. And in this case, what happens? So you have, this is something like a typical working memory experiment. So there, the animal is presented a cue, right? And you want the queue, usually in working memory, the queue gives you a surge of activity that lasts even when the queue is removed, right? So you expect that your firing frequency will become much larger than before the queue, even if the queue is removed. That's the duration of the working memory, right? And what happens without astrocytes, so when you only have excitatory and inhibitory neuron, at least in this simulation, that the blue, that when the queue here, when you have the queue, it increases the firing frequency of the neuron, but when you remove the queue, it's in blue, it gets back to the same level that before the queue. So there's no working memory. Now, if you add the gliotransmission in an astrocyte on top of that, that's the orange stuff, you see that the queue will increase the synaptic release probability of the astrocyte, and this will increase the frequency of firing of the neurons. This will give you the working memory that you are looking for. I let you look at the paper, but it's the same kind of simple model that I've just shown you before, at least in this case, in case of working memory. Okay, so now, to be more general, this is a kind of idea that people have now. So we have in mind a system where information can flow between neuron and astrocytes in this direction, for instance. So you have one neuron, when it fires there, the neuron 1 fires there, two dendrites, and the astrocyte in blue in the middle. So when it fires, the presynaptic will release glutamate. The glutamate will give rise to calcium surge in calcium signal in the astrocyte. In turn, this will, for this synapse, the same that has been activated gives you a bit of LTP. But the calcium signal can also reach another synapse of the same neuron and gives you LTP in a sort of heterosynaptic way, so in a synapse that has not been activated. And then the calcium signal can, even if the calcium signal is propagated in the astrocyte, give you a change in the transfer of information in the synapse, much more far away from the initial stuff. So that's the idea. Again, this is very nice to do nice drawing like that, but since we still don't know how the calcium signal, how far they are propagated, it remains purely schematic, right? And now, so to maybe you've heard some controversies about gliotransmission. In fact, it turns out that the experimental community that works in the astrocytes is currently, even it was the case 2 years ago, but even now, divided between two groups. The groups that are the glio believers, that people that are, I mean, they believe that gliotransmission, the release of a neurotransmitter by the astrocyte exists and that it works, it does something in vivo. And glioskeptics, the people that think that it doesn't exist in vivo, and that in fact exocytosis by astrocyte doesn't exist, and astrocytes don't release neurotransmitters. And those glioskeptics, in fact, it turns out that they give arguments that it's true that in several experiments where you will expect gliotransmission, so you will expect astrocyte to release glutamate or ATP, whatever, it's not observed. So in many cases, you expect the astrocyte to release transmitters and they don't release anything. And even more, if you look at in microscopy, if you have calcium-dependent release of vesicles by the astrocyte, then somehow you should see the vesicles. And in many cases, you know, in the experiment, under the microscopy, you don't see the vesicles. So that's why people believe that some people believe that it's an artifact, this gliotransmission. And maybe this can be a very recent paper again by Andrea Voctera. Maybe this paper can solve this controversy. In fact, in this paper, it's a typical single cell transcriptomic study. And so if you take hippocampus cells and you do a typical single cell RNA-seq, and you can cluster the cells. So you have glutamatergic neurons, GABAergic neurons, microglia, and so on. And there, this cluster is a cluster where you find the astrocytes. And now you can zoom into the astrocyte and do also the clustering. And then you see at least 7 clusters, 1, 2, 3, 4. That's what they found. And now, if you look at what they have in terms of gene expression, those clusters, you see clusters. For instance, the 7, the one that is here, that is the only one that has all the proteins and the mRNA that are required to do exocytosis, to do gliotransmission. So the conclusion is that probably you have at least 7 subtypes of astrocytes, and a single one, that's cluster 7, is the only one that does gliotransmission. So that's the important idea that is very important now, is that there's nothing like astrocyte. It's like neurons. Neurons, you have pyramidal neurons, interneurons, whatever. You have very different subtypes of neurons. Probably there are also very different subtypes of astrocytes, and only some of the subtypes are able to do gliotransmission, and some are not. I don't know if it's true. Again, it's a paper that's been published 2 months ago or 3 months ago, but maybe that explains why some people see gliotransmission, some others don't see it. Okay, so I will rush. Sorry, I'm late. So now this was all about calcium. I will just give you a couple of words about because it's important, non-calcium-based roles. So role for astrocytes that are not directly based on calcium signals. And the first of them is astrocyte role in homeostasis, right? Meaning the fact that astrocytes, one of the jobs of astrocytes is to keep the balance. Of course, when the presynaptic neuron will fire, it will release huge amount of potassium because the spikes are exiting potassium. And this potassium is toxic for the neurons. So yeah, it has to be removed. And the guys who does this is mostly the astrocytes. So you have this core 4.1 channels that I've already talked about that get the large amount of potassium from the extracellular, from the synapse, and push it intracellularly and remove it somewhere, for instance, in a blood vessel. So that's one important point. And the same for glutamate. If you have a large period of high firing rate from the presynaptic neuron, it will release huge amount of glutamate, and it has to be captured back. And it turns out that so you have, of course, glutamate transporter in the presynaptic elements, but in many synapses, the main glutamate transporters are in the astrocyte. So the main flux of glutamate out of the synapse, usually, most of the time, it's made by the astrocyte. And again, so it's transformed in the astrocyte from glutamate to glutamine, and glutamine is get back to the presynaptic neuron that takes it back to glutamate. And of course, those two uptakes are hugely important. And if you change them, you will change the synapse. And again, that's the example that I've shown you before, this paper with the depressed animals. What happened is that if you are in a normal site, the cells that are affected have a tonic firing. Now, the depressed animal, they have an overexpression of KER 4.1 channels there in the astrocyte. So because you have more channels, you have less potassium in the out of the neurons. And this is enough to change the neurons from tonic firing to bursting firing, right? So if you perturbate the concentration of potassium around the neuronal membrane, you have burst firing. And this is enough to change the animal, because in fact, those neurons, they inhibit the VTA that will they excite the VTA, sorry, that will inhibit the cells that produce dopamine. So when you are depressed, or when the animal is depressed, these cells will begin to be burst firing. This will have an increased activation of these neurons that will increasingly inhibit the dopamine producing cells. So you have less dopamine, and probably that's where the symptoms come from. So just my point there, that a small stuff like that, dysregulation of potassium uptake is enough to give symptoms like depression. And it's a bit the same for glutamate uptake. Just an example here. This is an example of STDP, so short-term dependent plasticity, in the striatum at the corticostriatal synapse. So the first synapse between the cortex, the pyramidal neuron in the cortex, and the medium spiny neurons in the striatum. And in this synapse, it's something that Laurent Venance in Paris has been working on for long. This synapse is very easy to do STDP, so spike timing dependent plasticity. So maybe you know that the spike timing dependent plasticity is just the idea that you take the presynaptic and the postsynaptic, and you stimulate pre, then post. You wait and you do it again. So post, pre-firing, post-firing, pre-firing, post-firing. You do it maybe 100 times. And then you change the order. And afterwards, you do post-firing, then pre. So you play on the difference between pre first, then post then, or post first and pre then. And what you see is that if you have a positive timing, delta t, that means that when you have first the post and then the pre firing, you will have a LTD, the synaptic rate will decrease. Whereas if you do the other way around there, when you do first the pre, then the post, pre, then post, pre, then post, then you will an increase of synaptic rate. So that's the an LTP. But this works only when the pre and the post, they are quite correlated, when the time between them is between 10 milliseconds and maybe 40 milliseconds. Right now, if you take much larger examples, time scales, for example, 50 milliseconds, 500 milliseconds, there it is a case where you do first the presynaptic stimulation, you wait for half a second, and you do the post then, and then it's too short, too long, and in this case, you have no plasticity at all. This is just a zoom out of this part, right? So this is a very interesting type of plasticity because it depends on the timings of your spike trains. Now, if you block the transporter, the glutamate transporter in the astrocyte, so what the DHK does, so you block the transport of glutamate from the synaptic cleft by the astrocyte, then that's the blue stuff that you have here, you completely lose the plasticity. That means that you have no LTP, and you have something that you have a plasticity even for very large time scale. So the function of your synapse in terms of timings has changed completely. And it's not, I'm sorry, it's a bit like a long list of stuff, but it's not only the expression of transporters, it's also the structure. And this is something that has been found maybe 20 years ago by Stéphane Olliette, is that the thing that, okay, if you have more transporter or less transporter, you will change the how many potassium is in your synapse or how many glutamate, and will change things. But it's not enough to do that. You only have to change the distance between the transporter and the glutamate, for instance. And how to do that? You just have to change the shape of your astrocyte locally, and you have your astrocyte that come much closer to the synapse and can even penetrate into the synaptic cleft. And when you do that, in fact, of course, your transporter will be much more efficient, and you will transport much more glutamate, so you will have much less glutamate in the cleft. And in fact, this kind of structural plasticity, the fact that the membrane of the astrocyte can be closer or further away from the cleft, is something that exists, and this regulates synaptic plasticity in the hippocampus as well. So it's not only the number or the concentration of transporter, it's also the structure of the synapse itself. And then this is something that has been discovered maybe only 10 years ago, is that it's not only that, it's that during development, astrocytes, so the branchlet, the perisynaptic astrocytic processes, the PAPs, to this part of the astrocyte that contact the synapse, they are the guys who do the synaptic pruning or consolidation during development. Meaning that you know that during development, the brain at first makes a huge number of synapses, and then these synapses, they are pruned. The ones that are active will be consolidated, and the others will be killed away, right? So initially during development, you make a huge number of synapses, and then you filter them out afterwards. So who does that? It turned out that this is made by the astrocytes. I don't want to go into the molecular details, but that what has been realized recently, that again during development, astrocytes select the synapses that must survive, and the ones that are too weak to survive are also removed by the astrocyte. So they are a way of selecting what synapses must stay and what synapses must be away. But interestingly, they do that for excitatory synapses, but also for inhibitory synapses. And so, and there's been last week in Washington during the SFN conference, there's been a very nice talk on that. If they control excitatory synapses and inhibitory synapses during development, maybe they control the balance between both. And you know that in the brain, at least in the cortex, you have this very important aspect that is the excitatory inhibitory balance between both. And maybe this balance is set during development by the astrocyte. At least that's what the people are looking for in the field currently. And then finally, and I will come back to something that Habib knows even better than me, is the astrocytic neuron lactate shuttle. The fact that, in fact, beyond metabolism, astrocytes give lactate to the neurons. It turns out that the neurons, they are very, very specialized cells, and they don't do many things. They are so specialized that the only thing they can do are spikes and making spikes. They don't even, in a way, they don't even know how to do their own food, right? Meaning that they need someone to give them food. And the guy that gives them food is the astrocyte. And there's a long controversy in literature whether what is the molecule that is given from the astrocyte to the neurons. It could be glucose. Glucose could be directly from the blood vessel to the neurons. But it's not sure. There's one part of the community that thinks that it's lactate, and there's a shuttling between lactate and glutamate, meaning that lactate could be, so in the end foot, the astrocyte could take glucose from the blood, change it into lactate intracellularly, transfer lactate to the neurons, and then the lactate there will be changed into pyruvate and go to the TCA cycle and to make new ATP. And that's also very important because it's essentially metabolism. In fact, it turned out that Pierre Magistretti in Lausanne has shown some years ago that this is also very important for plasticity, meaning that if you cut this circulation of this transfer of lactate from the astrocyte to the neurons, you cannot do, in many examples, you cannot do plasticity anymore. And in their case, it was because, in fact, the lactate that is transferred by astrocyte to the neuron is changed into pyruvate to go to the mitochondria, but it's also changed into NADH, and NADH could, in a way, activate NMDA receptor. So metabolism is not just for food; it's also something that is very important for plasticity and memory. So that's what. Well, I'm a bit long, but I just want to show this kind of thing that we did with Pierre Magistretti and Laurent last year, and we think, okay, so plasticity and metabolism is important for plasticity, but is it true for all types of plasticity? So what we did, we use in patch clamps, and Laurent did it. Laurent Venance did it in Paris. They use patch clamp to measure different forms of plasticity, and one form that is called theta burst, when you give a huge amount of stimulation to the neurons, very huge load, very large stimulation load, and we compare this with a protocol where you do a very low, much lower load of stimulation. So many stimulation there, small number of stimulation there. And then when Laurent looked at what is needed to express the stimulation. They saw that when you have a small number of stimulation, the plasticity can be expressed if you have glucose or lactate. Both are okay. Whereas if you have this kind of LTP, when you have a huge amount of stimulation, to express it, glucose is not enough. You have to have lactate coming from the astrocyte, or you won't express your plasticity. So that means, in a way, that you have two different types. I mean, if you have a small, something that is easy to do, you can do it on glucose, whereas when it is a huge amount of stimulation, you have to have lactate to do that. So we said, okay, is it the true as well for in behavior? So what Laurent did, he did exactly the same at the behavioral level. So they took rats, and they took the rats to do two tasks that have very different difficulty. The first one is the so-called novel object recognition, the NOR. And in this task, you have the animal sees the first day or the first days two obstacles, two objects, A and A, right? So it will the same ones, it will explore it. And then after 24 hours, you replace A, one of the objects, by another new object. And normal animals, they will get, after that, they will spend much more time around B because they will discover that it's a new object. They will realize it's a new object. And this is a very simple task. Now you can compare to a much more complex task. This can be done by rats and mice, for instance. And if you compare this with this task, which is the object in place task, this task can be done only with rats. The mice are not able to do that. And it's in this case, you have 4 objects from different types, A, B, C, D. So the rats learn that. And then after 24 hours, you swap the position of 2 objects, like A was there and C is there, and you swap them. And normal rats, they spend much more time around C and A than D and B because they learn that the position has moved, right? So 2 tasks with different, the same area in the brain, so again a hippocampus, but the 2 different difficulties. And then what they did, they removed glutamate lactate shuttle in the hippocampus of these animals. And what they observed is that this task that is very easy to do is okay, can work with glucose or lactate. The animal can do it even if it doesn't have lactate anymore. Whereas this one, the animal has to have lactate from the astrocyte to neurons to do it. If you remove the lactate shuttle from those animals, they are no more able to do the task. Okay, so it seems that there are 2 difficulties. So if you have a task that is hugely demanding, you have to have lactate, whereas if the task is easy, maybe glutamate is enough. So we did a model for that. I don't want to go into detail because I'm really late, but basically the model couple a presynaptic neuron, a postsynaptic neuron, the glucose, and in between you have a coupling between a metabolism, a model of the metabolism that we took from a model by Renaud Jolivet that he published 8 years ago, and we coupled it with a model for STDP. I want to go into the much detail. What is important for you is that the model is very, at least for metabolism, is similar to what you published in. I mean, you have been published this, working on this for several years already with Rennes.
This one is
yeah. But the important stuff is that when we made the model, of course, the first thing we did, we said, okay, we have an NADH there. So we try to see, is it possible that that is NADH that kills the LTP when you don't have lactate? And it didn't work. We couldn't have the model working. Whereas what we could do, and it's a bit weird, so I don't know, what was worried to do is to look at ATP in the neurons. And it turned out that if we consider that ATP below a certain fraction of ATP, LTP was not possible anymore. right? Meaning that if when you change, when we remove lactate, of course, you have much less ATP. And if ATP shuts down LTP, or at least if large ATP levels are required to express LTP, then with your model, all of a sudden, you can explain everything very easily. So what the model, the cues of the model is that probably it may be ATP that is the key actor deciding whether you can do a complex task or not. Again, I don't know if it's true. That's what the model says at least. Well, okay. Okay, and then to finish, almost to finish, two what I call uncharted territories. So two things that we still don't know. And the first one, I took it from our paper by Eilan Hu. In fact, so I told you, I remember this curve 4.1 that are overexpressed and cause depression. In fact, this curve 4.1, they were overexpressed in the astrocyte, but they were not, we didn't find them around the synapses. We found them around the cell body, the soma of the neurons. So in fact, it's true that there exists the astrocytes that not only contact the synapse, they also contact the cell body, the soma of the neurons. And that's where we find the overexpression of the curve 4.1. So those neuronal soma to astrocytic contacts, they exist, but nobody knows what they do. And it's very interesting because, of course, that's where the oxygen potential will be built, and that's where you can control excitability and so on. But it's nobody ever worked on that, but probably it's going to be important soon. And the last thing, of course, that's something that we are starting to work on. That is calcium dynamic in the NFIT, right? Of course, it's important because that's where lactate and so on comes from, and that's where your fMRI signal comes from. So if you want to do a multi-scale model from that explain from molecules to the fMRI, that's important that you have this kind of model. Turns out that it's just there are no good model of calcium signaling in NFIT. There's a model by very recently by Maria-Lina Linné in Finland and Alain Destex that is not very good, but I mean, it's a very good paper, but very simple. Probably we can do better. And even more, what is very important is that it turned out that these and food, quite often, they are coupled to the synapse. You see, this is an electron microscopy where you see this is the vessel. This is the NFIT. You see there the mitochondria, and you see that there in the NFIT, you have a synapse that comes there. A synapse with a presynaptic there, the vesicle and the postsynaptic. So you have something that is both an NFIT and both a perisynaptic tripartite synapse. So you have in a very small volume a coupling between NFIT and tripartite synapse, and probably that is a very important construction for the NFIT, but nobody ever worked. I mean, nobody currently understands what it does. And that's again something that we are currently trying to work on. Okay, so now I'm almost done. So now what I wanted to show you is that what is almost done now is the model of a single tripartite synapse, right? How the calcium goes and so on and so on. Now, how can we go from one synapse to a network of neurons and a network of astrocytes? Of course, you can say, okay, I have my synapses, and I choose some of them, and decide that they are tripartite synapses, okay? And there's no communication between the synapses. That's the first thing to do. But we know it's not true. The tripartite synapses, they are not isolated. They can do clusters, for instance. So that's why some of them, they can be clustered, and maybe they communicate, they share the calcium together. But how many do that? And how close do they share their calcium with the single cell they are from? Do they somehow add their calcium and share it with the astrocyte? And those astrocytes, do they communicate with each other when they are activated? Nobody knows. But so because nobody knows, it's difficult to do a network model, but we can still try. So we lack, that's what I say, we lack experimental knowledge to build. I think that's a really good multi-scale model. Not because we are not good, it's because we don't have the experimental data that will allow to do good work. But okay, we don't have that, but we still can do it. And the way to do it is to recycle, I would say, neuron-based model. And the first thing that, of course, that's something that you did, and Aurélie's PhD was 10 years ago, something like that. Started 2 years ago. Right. 2015, yeah. So for instance, this is an example you did for a neuronal mass model. So this is a typical neuronal mass, right? I want to go into detail, but you have two excitatory population, one inhibitory population, and then you can add gliotransmission. For instance, you can take into account the fact that some of the extracellular glutamate can be uptaken by the astrocyte, and then you can take into account as well that the fact that the threshold of the input-output of the sum of the population are regulated by extracellular glutamate and GABA. That's what you see here. Your threshold here is mu p, and it's a function of GABA and glutamate and in the extracellular. So of course, that's a way to use it. The nice with that is that you can do a bit of mathematics and you can do a bit of at least bifurcation analysis. But another thing that you can do is to say, okay, if you look in the input-output function of the astrocyte, right? So what we know is that the astrocytic calcium is a function, some function that we don't know, of the presynaptic rate, right? We don't know exactly what is this function phi there, but what we know that is positive. I mean, it's monotonically increasing, meaning that d phi increases when the rate of the presynaptic firing increases. But the thing that is very different from neurons is that this is true whether the presynaptic is glutamate or GABA. And that's very important, meaning that if the presynaptic neurons release GABA, you will have an increase in calcium, which is different from the classical GABAergic stimulation when the GABA decreases the excitation. It hyperpolarizes. You have no inhibition here. Glutamate or GABA from the neurons, they still increase the astrocytes. And now, if you measure glutamate release from the astrocyte, that was been done there, well, you see it spikes. You have random oscillation, and time to time you have spikes. So, well, it's very tempting to say, yeah, it's a bit like neurons, right? It's very similar. You have spikes, so because of the phi there, you have something that could look at the rectifying unit, right? So, like in neurons, you can say that maybe the astrocytic glutamate is 0, no release there, until the presynaptic rate reaches a threshold where you have this kind of glutamate admission. Of course, it's not precise, but that's the best we can do currently. The idea there will be to adapt neuronal models, classical neural models, to astrocytes, and do as if the astrocytes were working a bit like neurons. And that's what I will show you in the research presentation after the break. And I will stop here. And if you have questions, of course, or during the coffee break, we can discuss, of course, together. Thank you.